Hello, and thank you for joining us today for this LanternDx webinar. My name is Jason Amsbaugh, and I'll be the moderator today. In this webinar, Dr. Bhavika Patel and Dr. Brenna Dennison will present data on the development and application of multispectral imaging for the characterization of the tumor microenvironment and analysis of individual immune cell subsets pre- and post-therapy in breast cancer samples. During the presentation, if you have any questions, you can submit them using the Q&A button at the bottom of your screen. Please put your questions here and not in the chat window, so we can be sure to record your questions to answer during the Q&A session following the presentation. Also, we are recording this webinar, so if you miss any part of it, or if you want to rewatch it or share it with colleagues, you'll be notified in a few days by email that the video is available for on-demand viewing. Before we begin, we have a quick poll for our audience. So I'll go ahead and put these questions on screen, and please go ahead and answer them. The first question is, what is the biggest challenge in your research? What is the biggest challenge in accomplishing your research goals? Is it time, money, expertise, lack of resources, or something else? All right. The second question, what is the current stage of your research project? Are you working in discovery, preclinical, clinical, or some other aspect? The next question, which services might you be most interested in outsourcing? Histology, pathology, digital image analysis, clinical trial support, other, or none of the above? And the last question, short answer question, what is the primary research question that you are working on? All right, thank you for your answers to those questions. Without further ado, I'll turn this over to our first speaker, Dr. Bhavika Patel.
Good morning, everybody. My name is Bhavika Patel, and I'm a scientist at LanternDx. Thank you, Jason, for that great introduction. My colleague, Brenna Dennison, will also be speaking later in this webinar to discuss different approaches that we could do for multiplexing. But for right now, today, the topic of our webinar is using multiplex assays and image analysis to discover new insights in the tumor microenvironment. Before we begin, I'd like to give a little bit of background on our company. LanternDx is a CAP-accredited and CLIA-certified full-service clinical histopathology CRO. Our team is comprised of histotechnicians, scientists, MD and DVM pathologists, and analysts to provide guidance to clients when investigating and developing new biomarker strategies. Biomarker strategies are an important step in clinical work, which requires time, money, and resources. This is where our company comes in to aid clients in their preclinical and clinical work. Some of our services include routine histology services for both FFPE and OCT samples, biomarker discovery, clinical trial support, digital pathology, digital image analysis, and pathologist services that include adjudication. Today, the two services I will be discussing as part of our talk are biomarker discovery and digital image analysis, and how both of these are often combined together to help us dive deeper into understanding the tumor microenvironment. Going into a little bit more on biomarker discovery, we often see them used in preclinical and clinical settings. Prognostic biomarkers inform us about an outcome such as disease recurrence, disease progression, and death, regardless of the treatment that they are given. A biomarker is predictive if the treatment effect is different for biomarker positive patients compared to biomarker negative patients. Biomarkers are a widely used tool for both research and diagnostic applications. It can help expand our understanding of how drugs work, the efficacy of drugs, as well as guide clinical decisions. Several challenges exist with biomarker discovery. One of the biggest challenges of biomarker discovery is the vast number of samples that may be needed to assess all of the biomarkers that are of interest. In most cases, one biomarker does not fit all, meaning multiple biomarkers result in higher diagnostic accuracy than using a single biomarker. The most efficient way to assess a variety of biomarkers is by using multiplex technologies. Multiplexing is an efficient workflow to help identify biomarkers of interest. One advantage of multiplexing is it preserves spatial context, and we are able to assess all of the cellular phenotypes that are present within the tissue. In our lab, we utilize whole slide scanning, which is time efficient versus scanning specific regions of interest. Secondly, multiplexing allows for visualizing all of the biomarkers, whereas conventional immunohistochemistry usually only uses one biomarker. The ability to label multiple markers within a single tissue can provide valuable information about the complex tissue microenvironment. The composition of the tumor microenvironment varies between different types of tumors, but mainly it features complex immune phenotypes, stromal cells, blood vessels, and extracellular matrix. This image portrays a nice picture of all of the different components that may be a part of that tumor microenvironment, including macrophages, neutrophils, and dendritic cells that contribute to the innate immune response. Immune cells are a significant component of the tumor microenvironment and can either suppress tumor growth or promote it. Immune cells may also be predictive of disease prognosis, as well as provide guidance on how patients may react to certain therapies. Within the tumor, immune cells can be divided into two groups: tumor antagonizing and promoting, both of which play roles in how a tumor progresses. Tumor antagonizing cells are T cells, natural killer cells, dendritic cells, and a subset of macrophages and neutrophils. On the other hand, tumor promoting cells include regulatory T cells and myeloid-derived suppressor cells. Because of the various immune cells present within the microenvironment, understanding all these cell phenotypes present within the tissue can be valuable in developing strategies that aid in the treatment of cancer. Multiplexing gives us the power to visualize several of these biomarkers at once, enabling our researchers and clinicians to quickly interpret the complex phenotypes of the cells that are present within the tumor microenvironment, thus helping advance research discoveries. To begin the multiplex workflow, we start by identifying what cells are of interest and finding the best biomarker to identify those cell phenotypes. Next, each biomarker is optimized using a DAB chromogenic monoplex immunohistochemistry assay that best determines the conditions for that antibody. After finding the optimal conditions for each antibody, the next step involves using immunofluorescent fluorophores to ensure the same conditions from that chromogenic IHC can be applied. This step also involves testing biomarkers in various positions to ensure that the signal to background ratio is optimal through the multiple antigen retrieval steps during a panel. Lastly, using the information learned from the immunofluorescent monoplex optimization, we strategically place each biomarker into an appropriate position within the panel to ensure that we receive the best signal to background ratio. Oftentimes, this step may take a few weeks as the combinations of antibody placement varies with the number of biomarkers in the panel. Multiplexing can provide a user many data endpoints if analyzed using a powerful system. In our lab, we use the Visiopharm platform, which integrates deep learning to image analysis. Listed are just some of the endpoints we are able to output, including percent positivity for cells or percent positivity by area, co-localization of cells, H-score analysis, cellular morphology, such as obtaining measurements of cells, and subcellular analysis that allows us to analyze specific areas such as tumor and stroma, phenotyping analysis, and data visualization that helps us QC our data. By using an AI-driven platform, we are able to obtain accurate and reproducible results. Now that we have reviewed biomarker discovery, the importance of multiplex assays, as well as how a multiplex panel is developed, and the various endpoints we can obtain from multiplex assays, We will now discuss our results from an internal study that used breast cancer patient specimens treated with neoadjuvant chemotherapy using a 6-plex immunofluorescence assay with clinically relevant immuno-oncology biomarkers. Breast cancer is a common cancer diagnosed in females in the United States. Males can also be diagnosed with breast cancer, although it is a much more rare occurrence than females. One of the first steps is to conduct receptor testing on patient biopsies. Therefore, the clinicians can subtype patient breast cancers into different groups. These include estrogen receptor positive, progesterone receptor positive, human epidermal growth factor type 2, also known as HER2 receptor positive, triple positive, and triple negative. Triple negative breast cancer is the most aggressive form of breast cancer since drugs targeting receptors do not work. Triple negative breast cancer also grows quickly and has a high rate of recurrence, making it very aggressive. It can be treated using chemotherapy and immunotherapy, whereas heterogeneous tumors can be treated using hormone therapy and/or chemotherapy, as well as a combination. Our study analyzed breast cancer biopsies from 4 distinct patients. All patients had ductal cancer. Patients A through C were HER2 negative and estrogen receptor and progesterone receptor positive, whereas patient D had an aggressive form of triple negative breast cancer. All patient biopsies were stained using 6 biomarkers to help us understand how the tumor microenvironment and immune cell profiles change before and after treatments. CD8 was used to identify T lymphocytes, PD-L1 was used to stain the ligand for programmed cell death protein, FOXP3 identified the regulatory T cells, CD163 stained macrophages, PanCK identified all tumor cells, and CD4 was used to identify the immune cell population. After staining, all samples were imaged using the PhenoImager Fusion scanner from Akoya Biosciences. This scanner allows for whole slide imaging using 6 colors plus DAPI, which is used to stain the cell nuclei. One advantage of multiplexing is the ability to visualize multiple biomarkers within the entire slide. For this study, we analyzed only tumor regions from the biopsies. This chart summarizes the biomarkers and the color it appears on the image and what the biomarker stands for. On the right-hand side, this top row on this side shows the unique patient's pre-treatment samples, and the bottom row shows the respective post-treatment sample. Multispectral imaging of the whole slide allows us to image the entire biopsy to ensure all tumor areas are accurately analyzed. After scanning our images, our analysts use the Akoya Inform software to correct for autofluorescence. Images are then imported into the VisioFarm platform for further analysis. Using AI and deep learning, cells are segmented into nuclear regions, highlighted in blue here, and cytoplasm, highlighted in white. We then utilize the Phenoplex guided workflow to easily phenotype cells by adjusting thresholds for each biomarker individually to determine cell positivity. In this example, small colored dots seen in this image help represent individual phenotypes that are present within the tissue. Each phenotype would get a different color dot associated with it. By phenotyping cells, we are able to better understand the tumor microenvironment. At a quick glance, phenotyping analysis is able to output a variety of information, including information about each individual cell. Our analysis focused on the number of cells that express specific phenotype. The first step is to use the Phenoplex guided workflow to identify all phenotypes. In this example, we're going to use patient A, who was treated using hormone-based chemotherapy and was a HER2 negative patient. This patient had 39 unique cell phenotypes. One advantage of the Phenoplex guided workflow is it makes finding phenotypes easy by setting thresholds for each individual biomarker that helps determine the cellular positivity. Phenoplex generated a color-coded list of all the phenotypes and outputs the raw information. For this patient, the 39 unique cell phenotypes included single cells seen in the top left-hand corner, dual positive cells seen in the bottom left-hand corner, triple positive cells seen on the upper right-hand corner, and quadruple cell phenotypes seen on the bottom right-hand corner. For this presentation, we will focus on only a select few phenotypes that were relevant for this assay. That includes pan-CK, FOXP3, FOXP3 and CD4, CD4 and CD8 positive cells. First, our most prominent observation was the decrease in pan-CK expression. Pan-CK is a general tumor stem cell marker. Although pan-CK is not a target during therapy, it is commonly used to quantify the tumor cells that are present. Patient A was a 66-year-old with ductal cancer and a HER2-negative tumor that was treated using hormone-based chemotherapy followed by radiation. Here, we see one field of view region for the pre-treatment biopsy on the left-hand side. All pan-CK positive cells, including those that are dual positive with another biomarker, are labeled with the colored dot in this image. The post-treatment shows a decrease in the number of these dots, which suggests a lower number of pan-CK positive cells. When this data was analyzed, we see these qualitative results do indeed show a decrease in the number of tumor cells that are present. Specifically, this patient showed a 12% decrease in pan-CK positive cells post-neoadjuvant chemotherapy that may suggest we have less tumor cells overall. Similarly to patient A, patient C was an 81-year-old with ductal cancer and also a HER2-negative tumor. She was treated using tamoxifen, but no radiation or chemotherapy. Tamoxifen is a hormone-based therapy that is commonly used to treat receptor-positive breast cancer and can help stop cancer cells from using estrogen and progesterone to grow and spread. Similar to the previous patient, we see many pan-CK positive cells indicating tumor cells throughout this biopsy. However, post-treatment, the tumor cells significantly decreased from 70% to 44%, indicating that the tamoxifen did greatly impact this patient in a positive way, as well as post-treatment after receiving the neoadjuvant chemotherapy, it helped reduce the number of tumor cells that were present. Lastly, patient D also showed a significant decrease in pan-CK positive cells. As a reminder, this is our triple-negative breast cancer patient treated with radiation followed by chemoimmunotherapy. The pre-treatment biopsy shows many pan-CK positive cells indicated by the red dots on this image. These results suggest that although this tumor was quite aggressive, we see a decrease in the number of tumor cells post-treatment. Also, what is interesting about patient D is it had almost 50% reduction in pan-CK intensity post-treatment compared to patients A and C, which were HER2-negative tumors. Patient D had triple-negative breast cancer and was treated with immunotherapy. Therefore, we can speculate that this drastic change could be related to the immunotherapy treatment. The post-treatment shows some pan-CK positive cells on this corner, but it also shows this interesting cluster of immune cells within the tumor. We've investigated these cells across all patients and saw there is an increase in T cells post-treatment for patients A, C, and D. CD4 is a glycoprotein that is found on the surface of immune cells, such as T helper cells, monocytes, macrophages, and dendritic cells. CD4 positive cells are known to help the immune response against infection and disease. In our study, we see that CD4 increases in patients A, C, and D, which suggests that there may be a recruitment of histiocytes and T cells post-neoadjuvant chemotherapy. However, patient B shows a decrease in T cells. This data does need to be expanded in order to include more biopsies to make any further conclusions. However, the results right now indicate that patient B may not be responding well to the treatment as the T cells have decreased. Furthermore, CD8 shows an increase in patients A, C, and D, which also suggests a good prognosis for these patients, indicating their treatments may be working effectively in treating the tumor. However, we see again that patient B shows the opposite trend, which may indicate a poor prognosis. Again, more biopsies would need to be analyzed, but the main takeaway from this is that the fact that we were able to use the same multiplex panel to stain all 4 patient biopsies and investigate some prominent immune phenotypes present. Overall, an increase in CD4 and CD8 cells indicate a patient may have a better immune system response and suggesting that their immune system may be able to effectively decrease the size of their tumors. Lastly, we investigated FOXP3 because research has shown that this is often overexpressed in breast cancer and may be an indicative marker for how aggressive that cancer is. In our study, we see a decrease in FOXP3 cells for patients A, B, and C, suggesting that the tumor may be responding to treatment. FOXP3 and CD4 dual positive cells increased in patient D, which may suggest recruitment of T helper cells post-treatment. Patient D had triple-negative breast cancer, which may be the reason why FOXP3 is elevated in their post-treatment as their cancer is more aggressive. Again, we need additional biopsies to make any further conclusions for these patients. In summary, the 6-plex panel consisting of FOXP3, PD-L1, pan-CK, CD4, CD8, and CD163 biomarkers was used to analyze the immune microenvironment of pre- and post-breast cancer patient specimens. Specifically, this panel helped us visualize the T helper cells and tumor cells in order for us to better understand the immune response for these patients. We observed a decrease in pan-CK in patients A, C, and D. Patient B showed an increase in pan-CK positive cells. However, we can't make any conclusions based off of this just due to the sample size. In order to make any definitive conclusions, we would want to repeat these findings using additional biopsies. Second, we observed there was an increase in CD8 and CD4 positive cells, both of which suggest recruitment of T cells in order to help fight against the tumor. This was seen in patients A, B, and C. However, patient D did not show this trend, which could be due to their tumor being more aggressive. We also observed that FOXP3 decreased in a subset of patients, indicating those patients may be responding to their treatments better. Overall, cellular phenotypes change between patients as each patient receives different treatments. Our panel was utilized in order to better understand the T helper cells and tumor cells within the microenvironment. Multiplex panels, such as the ones that I just presented, may help scientists, researchers, and clinicians make informed decisions on patient care, as well as provide more individualized treatment plans. My colleague Brenna will now discuss how multiplexing can be expanded to RNA and how a dual in situ hybridization and immunofluorescence assay could help expand our multiplexing needs.
Good morning, and as mentioned, I will be discussing the use of multiplexing with in situ hybridization, or ISH. Fluorescent and chromogenic ISH can be used for identifying specific nucleic acid targets. FISH can be used to identify copy number variations like deletions and amplifications. It can also identify translocations and extra chromosomal DNA. KISH can also be used to study gene expression signatures at the cellular level with spatial and single cell resolution. Utilizing the same workflow discussed earlier, another project that we worked on was creating a hybrid multiplex ISH and IF assay. This hybrid multiplex panel can provide insights into the visualization of spatial organization of the tumor microenvironment. It also enables semi-quantitative assessments for the expression levels of cytokines and their relative expression within tumor and immune cells. Cytokines have a large impact on the tumor microenvironment by affecting the cell growth, survival, inflammation, and differentiation. This hybrid multiplex panel can help to correlate response to treatment, especially with checkpoint inhibitors. So for this study, we used our in-house control box for non-small cell lung cancer, or NSCLC, and breast cancer tissue to optimize this method. We analyzed the tumor microenvironment of these tissues with a panel for the cytokine RNA and immuno-oncology, or IO, protein biomarkers using FISH and IF. These tissues were stained using ACD RNA-seq probes for TNF-alpha, combined with protein antibodies for CD68 and CD163 IO protein biomarkers. The staining was performed on the Leica Bond RX automated stainer using an optimized protocol developed by LanternDx. And like previously discussed, the multispectral whole slide imaging was performed on PhenoImager Fusion, and phenotyping analysis was performed using the Visiopharm software. So here we have unmixed multispectral scans of the NSCLC tissue on top and the breast cancer tissue on the bottom. And these were stained with the ISH-IF panel. These are zoomed in 4DX images that show the TNF-alpha RNA-ish cytokine in yellow with Opal 570, as well as the IF biomarkers CD68 in green with Opal 520, and CD163 in orange with Opal 620. We then performed our image analysis workflow, and this is where the nuclei was detected using machine learning, then phenotyped by the underlying nuclear and cytoplasmic signal. Here I'm also showing detailed 4DX view of the NSCLC sample on the left-hand side. And on the bottom, or on the top, we have this without the labels, and on the bottom, we have the phenotype labels. And then on the right, I have the breast cancer tissue, again, with the labels on the bottom and without the labels on the top. On the right there is a key showing the different color labels that are associated with the various cell phenotypes. The cell phenotyping percentages were then graphed, and that is shown here on the slide. NSCLC is on the left-hand side, and the breast cancer is on the right. Again, a key is underneath, and you can see this shows the color labels that are associated with each phenotype that matches for each bar. Both these tissues had the highest percentage of cells with TNF-alpha expression phenotype, with the NSCLC showing higher expression levels. So our hybrid multiplex ISH and IF protocol was optimized to analyze the tumor microenvironment of NSCLC and breast cancer tissues. This enabled us to simultaneously detect the RNA of cytokines and the protein of specific IO biomarkers. CD163 positive macrophages were seen predominantly within the tumors, whereas the CD68 positive macrophages were identified towards the periphery in the non-tumor microenvironment. NSCLC tumor cells showed higher expression levels of TNF-alpha compared to breast cancer tumor cells. This multiplexing enabled assessment of the relative expression levels of TNF-alpha in the immune cells and tumor cells. So in conclusion, we can utilize multiplex assays to help further our understanding of the immune microenvironment and its phenotypic variations. These multiplex assays allow for multiple biomarkers to be visualized on a single slide, therefore using less tissue. We can combine RNA and protein detection within the spatial environment of the tumor, thus allowing us to detect changes in the immune cell phenotypes and cytokine expression levels and patterns. This can also be expanded using DNA probes. Digital image analysis is a powerful tool that can aid in research and clinical settings and allows for rapid turnaround and higher throughput analysis. Thank you for your time this morning, and we're happy to take any questions. Thank you.
Thank you, Brenna. And thank you, Bhavika, as well, for your guys' presentations. So quick reminder, if you have questions, please go ahead and submit them using the Q&A button here in your Zoom toolbar, and then we'll answer them as we go. All right. So here's a couple questions kind of coming in. The first is, can you detect surface markers and intracellular markers at the same time?
Yes, co-localization is one of the benefits of multiplexing, and the guided Phenoplex workflow that we utilize from Visiopharm actually makes this much easier. Before, we used to have to, only one label could be used to identify a cell. So if that cell was only pan-CK positive, that is the only label it would be getting. But now with this advanced workflow, we're able to label that same cell with multiple other biomarkers, making this co-localization analysis very simple.
Excellent, thank you. The next question is, can multiplex immunofluorescence be used as a diagnostic tool?
Great question. So it can't be used right now as a diagnostic tool, but I do think that it is heading that way. Multiple laboratories have utilized multiplex immunofluorescence to report prognostic or even predictive values of immune subsets in several types of cancers, but it hasn't been used in clinical settings thus far. One of the reasons, I think it's because it's really difficult to validate an assay. There's lots of moving parts to it. There's different protocols that may be used across different labs. And one of the hardest things is that the data must be analyzed using digital software. It's really difficult for a pathologist to score a multiplex assay. It would take a long time. And so until the field kind of validates a process for digital image analysis, I think it's heading that way, but it might just take a couple more years before we're able to do that.
Great, thank you. How many biomarkers can you target per sample? I guess, is there a plexing limit to a multiplex assay?
There is not. It depends on the instrument that we have. So currently in-house, we can multiplex 6 biomarkers and then include DAPI. And so that's just with the PhenoImager Fusion, we have 6 different color options and then including DAPI. So that's for immunofluorescent assays.
Okay. Question here, can bone marrow samples be used for these types of assays? Or do you have any experience with that?
Yes, bone marrow samples can be used. We have some bone marrow that we have tested at just simple 2-plex, 3-plex assays on. We have seen good results with it. And higher plex, it would just depend on what sort of questions are, like someone is asking. We can definitely optimize a panel. We do have ready to use panels that we offer. And one of the good things about those is, the panel has already went through the development phase. And when we're trying to apply it to a different tissue, the optimization process definitely becomes slower. So we can, adjust some settings to make it work across multiple different tissue types.
Gotcha. And I guess maybe a follow-up question on that. Have you guys tested the panel that you presented on other tumor types beyond breast cancer and non-small cell?
Yes. So this particular panel that we introduced earlier, we've only used it on breast cancer, but another panel that we've developed, we've used it across breast cancer, colon cancer, non-small cell lung cancer, head and neck cancer. And so one of the great things about multiplexing panels, like I said, was, we can reproduce that panel among different tissue types, and it just requires a little bit of optimization on our end, but not as difficult as it may seem. So panels can be very versatile and can be used across multiple tissue indications.
Thank you. Question here about mouse tissue. Have you had good results or any issues using frozen mouse brain tissues for multiplex assays?
So OCT samples are a little bit more difficult for multiplexing because the epitopes may be damaged in the process. We have used FFPE mouse samples for multiplexing from, I think, between 4 to 5 plex assays. So we have used mouse tissues. Brain, we have one panel that we worked with Akoya on, and so that one we do have some good results as well.
Perfect, thank you. Just a general question here. Do you tend to see a lot of background during multiplexing?
So this really depends on how strong the antibody is. And so one of the things is, what we do in our lab is when we have a panel that has been optimized, we do perform an autofluorescence slide. So that slide is stained with all of the reagents minus the antibody and the fluorophores. And this gives a good indication on how fluorescent the tissue itself is. And then we use the Akoya Inform software, and we can correct for that autofluorescence by selecting the most autofluorescent region in the naive tissue, and then going into the sample tissues and subtracting it. So you can see background, but we do a good job at making sure our quality controls can subtract it.
Gotcha, thank you. It looks like we just have one question left in the queue. And so any attendees, if you guys have any other questions, please put them in. This question is, how do you have confidence in digital image analysis when manual scoring by a pathologist is considered the gold standard?
Yeah, so all of our IA is guided by pathologists from selecting which samples to use for the training, as well as to assigning any thresholds for each biomarker. So we also have the pathologist QC the final data to ensure that it makes sense. And then if not, we do go back and work with the pathologist to update that app and make adjustments as necessary.
Gotcha. Okay, the next question is, how long does it take to create a new 4 to 6 marker panel?
Yeah, great question. So it depends on if those antibodies in the panel have been optimized via a chromogenic IHC. So one of the first steps for our lab is that we make sure that each antibody does have an optimal condition using just the gold standard DAB staining. And anywhere between 1 to 2 weeks we can spend for each antibody, and we can do that consecutively since the antibodies do not rely on one another. After that, once that step is done, we can complete the IF monoplex in probably another 1 to 2 weeks. And then the panel, I would say maybe another 3 weeks. So I would say in less than 8 weeks, we would definitely have a 4 to 6 marker panel developed.
Excellent, thank you. All right. It looks like that's all the questions we have in the queue. If you do have any additional questions, or you'd like to connect with the speakers today, you can reach them at info@lanterndx.com, or of course, visit their website. Also wanted to remind you that the webinar was recorded and it will be available for on-demand recording in the next few days. And again, we'll send out an email when that's available, so you can share it with your colleagues, or if you missed or want to rewatch any part of this webinar. Stay tuned for more webinars to come, and please subscribe to our email list to stay up to date on how the breakthroughs in multiplex imaging are advancing clinical and translational research. I'd like to thank our speakers again, Bhavika, Brenna from LanternDx, and thank you everybody for attending today's webinar. Goodbye.